Hi, Michelle Glass here. Welcome to our final episode of our Chapter 23, Lecture 23 series. We've been spending all of this chapter discussing the respiratory system, and our job here is to discuss how that system is regulated. There are actually several local types of regulations to the respiratory system. One we've talked about extensively already. So we had a whole talk on hemoglobin saturation and how that can be affected by factors such as partial pressure of oxygen, temperature, and pH. Some of those factors, or all three of those, are, really seen different in active tissues versus resting tissues. Also, we will see that levels of carbon dioxide and oxygen can change the diameter of both blood vessels and airways. So looking at our peripheral arterioles and capillaries, when you have a high partial pressure of CO2, those vessels are going to be dilated so that you're increasing your blood flow there. So if you have a high partial pressure of CO2, then that means you need to increase your blood flow to get rid of that CO2 and to bring oxygen to those tissues. In our alveolar capillaries, so now we're located in our lungs, we see that they are actually constricted when our levels of oxygen are low. So what that means is when we have high partial pressure of oxygen, those capillaries are dilated, so we have good blood flow when we have plenty of oxygen close to those capillaries. And then we'll see that our bronchioles will increase in diameter when you have a buildup of carbon dioxide gas. So when you have a high partial pressure of CO2, those airways are going to be larger, so you're going to get more airflow into the respiratory portion of the lungs in order to increase your gas exchange. So this is pretty interesting to see how our gas levels can control the diameters of different blood vessels and airways to increase blood flow or increase airflow as needed. Our remaining topics are dealing with our nervous system controls. We do see that we have controls at 3 different levels. Some of these levels are unconscious regulation. You already know that. We don't have to think every time we need to take a breath. And some of those levels are going to be conscious control, and you know that too. We manipulated our breath as part of our conversation earlier in this chapter. The first level is going to be the most primitive level, and this is described as our respiratory rhythmicity centers. So we have 2 different collections of neurons that are going to be involved in coordinating our inhales and our exhales. These are located at the bottommost part of the brainstem, so the most primitive part, the medulla oblongata. Controlling these centers, you have a second level, and we can talk about this level as a group of respiratory nuclei. So nuclei is our term of neurons that are, basically a processing center. Your respiratory nuclei are located at the pons, which is moving up on the brainstem, but still the brainstem, so it's still fairly primitive. We're still here talking about autonomic nervous system control, meaning unconscious level processing. The conscious level, the third level, the higher centers, can override both level 2 and level 3. Level 2 and level 1. Here we see that we have collections of neurons in the cerebral cortex, the limbic system, and the hypothalamus that can then make conscious controls of levels 2 and level 1, giving us conscious control of our breath. As we look at our level 1, we have the respiratory rhythmicity centers. So this is setting our basic respiratory rate, our rhythm of inhales and exhales. Now we have 2 different collections of neurons. We have one on the back of the medulla oblongata. So this group is called the dorsal respiratory group, and this is abbreviated DRG. These neurons control your quiet inhale. So remember, your inhale is triggered by the contraction of your diaphragm and external intercostals. When they contract, the rib cage lifts and the abdominal cavity gets compressed down, increasing the volume of your lungs, decreasing the pressure, pulling air in. So that's our inhale. Then we see that stimulation of that muscle stops. That muscle is allowed to relax. That's going to compress your rib cage, compress your lungs, decreasing volume, increasing pressure, pushing air out. So remember, we have our passive exhale here. You might use your D to help you remember. The DRG controls the diaphragm, which you should already know is part of our quiet inspiratory muscle, okay? Now, one of the triggers to tell the DRG to stop contracting is a reflex that's known as the inflation reflex. You have a group of special cells in the lungs, which are called stretch receptor cells. Stretch receptor cells are stimulated as the lungs expand. We need to prevent overexpansion of the lung. So as the lung is getting larger, then those stretch receptors are stimulated. That's going to actually send a message to the DRG to stop stimulating the diaphragm and external intercostals, letting those muscles relax, giving you an end to your inhalation and a start to your passive exhale. In some cases, you might also initiate the VRG, which is our next group of neurons. The VRG is located at the front of the medulla oblongata, and so it's described as the ventral respiratory group. The VRG is going to be stimulated when there is an increase in the DRG activity. So when you're starting to get increase in your respirations, then you're going to start moving into what we've called the forced breathing, where you're going to recruit those accessory muscles. You might start by recruiting all of your inspiratory muscles, which is going to give you that forced inhale. And then you're going to stop stimulating those muscles, start stimulating the accessory expiratory muscles, giving you that forced exhale. And then you're going to just keep moving through that cycle. The VRG can be coordinated by the deflation reflex. The inflation reflex is involved in your quiet inhales as well as your forced inhales. The deflation reflex is going to be part of your forced exhalation. In this case, as you're starting to compress the lung, you don't want to decrease or deflate the lung too much. So as that lung is getting smaller and smaller volume, you're going to stop stimulating the expiratory centers, and you're going to start stimulating the inspiratory centers. So that's how we're going to keep this cycle going at the VRG. Both of these reflexes are initiated by stretch receptor cells. The second level is located at our pons. You have another group of neurons that are going to help regulate the depth and the rate of the respirations. Now remember, level 2 is going to synapse and control level 1. And we have two different groups of cells here. So we have one group of cells that's part of what's called the apneustic center. The apneustic center is going to help increase the intensity of your quiet inhalation or your quiet breathing. And so we'll see that this group of neurons is going to synapse onto the DRG and control that quiet inhale. Now remember, our quiet inhale is stimulating diaphragm and external intercostals to increase the size of our thoracic cage, increasing our lungs, pulling air in. And then somehow we have to stop that signal so that those muscles relax and our lung volume decreases and the air gets pushed out. It's the second group of neurons that is going to help with that passive exhalation. The pneumotaxic center is going to inhibit the apneustic, If the apneustic center is inhibited, then we're no longer signaling to the DRG to cause the diaphragm and external intercostals to contract. So this leads us to our passive exhale. Those muscles start relaxing. The thoracic cage is going down in size, the lungs are going down in size, and air is getting pushed out. The pneumotaxic centers are not only involved in our passive exhale, they are also involved, or it is also involved in our forced exhale. So we can see pneumotaxic centers also controlling and signaling exhale at the VRG. And then, of course, having the ability to override both levels that we've discussed so far. You have your level 3. This is your higher order centers. This is your conscious level processing. This is located in the cerebral cortex, the limbic system, which is our motivation and emotional center, our hypothalamus, which has a strong link with our endocrine system. Level 3 is conscious control. So we know that our thoughts and our emotions can either, increase our respirations or decrease our respirations. We can then, through thoughts and emotions, trigger a sympathetic response or a parasympathetic response. Also, in some of our exercise, we need to control our respirations. If you're swimming, you have to have a coordinated time to take a breath and then hold your breath. And so we'll see level 3 helping to control that as well. Obviously, all of this control centers is responding to some kind of stimulation from the body. So we have different types of stimuli which are being used to help interpret when to inhale and when to exhale. We've already talked about the lung inflation and deflation reflexes. Blood pressure is discussed for us back in chapter 21, so we didn't spend a whole lot of time making that connection, so we'll do that a little bit here. And then we'll focus more on the homeostatic mechanism for regulating our carbon dioxide gas levels and then connecting in with that our pH levels. When we look at our blood pressure and our respiration rates, we should notice that there's an inverse relationship. If your blood pressure is low, then that means you're not getting adequate perfusion to your tissues. So that means you get a buildup of CO2 at your tissue level and you're not getting enough oxygen gas there. So of course we need to increase our respirations so that we're getting more exchange with our environment, we're bringing more oxygen into the body, and we're getting rid of our CO2. Blood pressure is detected by a group of cells called baroreceptors. Those are receptors that are detecting differences in blood pressure. Our chemoreceptor cells are going to be detecting differences in both pH and carbon dioxide gas levels. Now remember, these two things are linked. Carbon dioxide gas, we already have talked about, combines with water to make carbonic acid. Acid is a weak acid, but it will lower your pH. So if you have a high CO2 level, you're going to have a low pH, meaning that your solution is more acidic. So these two things are connected in together. We see a direct relationship between CO2 and respiration rates. So when CO2 levels are high in the body, then we increase our respiration rates in order to get that exchange to happen. Because there's an inverse relationship between CO2 and your pH, based on the way the pH scale is described, then when your pH levels are low, that means it's acidic, then your respiration rate is going to be high. Let's look at the negative feedback mechanism. How does the body control when you have elevated CO2 levels? Elevated CO2 levels are described as hypercapnia. So this can happen if you're not getting good exchange with your atmosphere. So if you have slowed down your respiration rate, if your respirations are very shallow, if you're holding your breath, you're going to have a buildup of CO2 in the body. And essentially, we're just going to increase our respiration rate in order to get that CO2 out to the atmosphere. We're going to see chemoreceptors are stimulated by elevated CO2 level. Those chemoreceptors are located in our arteries and the medulla oblongata. So this is going to stimulate our VRG for our forced exhale. Your expiratory muscles are stimulated, and inspiratory as well. You're overall increasing your respiratory rate and your depth of respirations, increasing your exchange with the atmosphere. So you're getting rid of that extra CO2 and also bringing in some oxygen. So CO2 levels should decline, going back to homeostatic normal. So it's pretty straightforward. We see essentially the same thing happening, but in reverse when our CO2 levels are too low. CO2 levels that are too low, this is referred to as hypocapnia. This might happen in the case of hyperventilation. So if you've increased your respiration rate and you've been pushing out a lot of CO2, then you might end up with CO2 levels that are too small. And so essentially, we just need to slow down our respirations in order to retain some of that CO2 in the body. So it's basically the reverse. So again, we have chemoreceptors in the same locations, detecting our low CO2 levels. Now we're going to inhibit our respiratory muscles so that we're slowing down our respiration rate. And so that's going to lead to an increase in our CO2 levels. So pretty easy. Okay, guys, that is it for our respiratory system.